Welcome back. Okay, one last time. Anybody has any issue installing the previous tools? Anyone? Or any questions or any doubts till now? No? Okay. Then great. Okay, Hadi, that's good. So then it means everything is fine. Then let's move on to some more stuff. Yeah. Just give me a second. Let me share my slides. Oh, but maybe before I share my slides, I'll do one thing. I'll upload the slides also in the in Google Drive. Can everybody just ensure that they have downloaded it? I'll just upload it in a second. Because you'll have to copy some of the commands from here. I'll let you know once it's ready, okay? In the meantime, you can open the link to the drive where you have the study material. Yep, the file should be there now. Can I request everybody to please download it? Okay, can I get a thumbs up if you have done it? Or let's say the other way, and thumbs down if you're still doing it. Yes, Pratima, session 4. Okay, done, Pratima? Anybody else here left? Okay, if not, then we move on. Let me share my screen first. Can anyone confirm if they're able to see the screen? Anyone? Great. Okay, fine. So welcome back, everyone, once again. So now we would be moving on to the next session. So, what, okay, again, very quick recap of what we have done till now, right? So we have first of all learned, we have installed a Linux kind of environment in our operating system. So that's WSL. If you have Windows, and for Mac, we already had that. And then we installed Conda, which is used for installing package environments and stuff. And before that, we learned how to navigate through the different file system, open files, folders, delete them, and open them, edit them, make a shell script and run it, right? And also we installed a few tools using Conda, okay? Now, we'll move on to a little bit. So today I will be talking about in this session a few things. So for example, first I'll take you through some of the standard file formats that we use in bioinformatics. And then we will download one data set from one of the public repository, okay? And then we will, work through that a little bit. We will do some sort of a QC. We will also do some sort of a trimming as well. So if you remember in the RNA-seq analysis workflow, some of the steps we will be following today, okay? And depending upon how far we go, we'll see how much we cover in this session, and the rest we'll, try to take it in the next one, okay? So the first thing, yeah. So further, one of the first sequencing formats. So even before this, right? So in bioinformatics, or let's say even in computation field as well. So we have sometimes files which are of a certain format, right? So for example, you sometimes have files in your system which can only open in Microsoft Word, some only in Excel, some in PowerPoint, etc., etc. Yeah. That is because the file structure or how the information is stored in those files is in a very particular format, and only certain applications can do that. Yeah. So having such standard formats is very common and important across more or less all domains. All right. And so bioinformatics is also, biology is not far away from this as well, right? So similarly in this, we have some of the very standard formats, which I'll be covering some of them right now. Okay. So the first one, which is the most basic format in biology, is something called as a one of the sequencing file format is a FASTA format. Okay. So what this FASTA format comprises of is two components. Yeah. So first of all, it is a very text file kind of format. Okay. So you would open this if you have to open it in your Word in your Windows system, you would open it in your Notepad. Okay. And in Mac, you would open it in something called as a text edit if you have. Or you can also open all of this in your VI editors as well. So you know how we open the VI files, right? For the shell scripts and stuff. Similarly, you can use VI to open and edit the files also. Yeah. So this, but what this format comprises of, so this format is used to store any kind of nucleotide or protein sequence. Okay. So so this sequence in terms of protein or nucleotide can be just, let's say, an amino acid sequence, or this sequence can be let's say DNA from anywhere. It could be genomic DNA, or let's say cDNA or mRNA or CDS sequence, anything for that matter. Okay. So those you use, so these file format FASTA is used to store such sequence related files. Okay. And this file, the way it stores this data is in the following format, where every sequence is stored using two different entities. So first is a header, which starts with a greater than sign, followed by whatever is the ID or name of the sequence, which is in this next line. Okay. So the first line starts with a greater than, and it contains the ID, and it can have as much details as needed. So for example, in this particular case, we have NC0006.12. So this 0006 is basically chromosome 6 from the human genome. And this particular sequence is from this location, that is about 151 MB to 152 MB of the human genome. Okay. Yeah, sorry, just 1 second. Yeah. Yeah. Okay. So, and like this in the header, you have all, you can ideally store all the information related to the sequence that you need. But most important one is the ID of the sequence. Yeah. So in this case, it is chromosome name, colon. And so this ID is coming from NCBI, which is one of the most common database. So a quick question here. Is NCBI database already covered in your lectures? So first of all, answer from the secondary students. So we are second and third year students, right? So first, for answers from secondary students, is this already covered? NCBI? No. Okay. Third year students? Is NCBI covered in your curriculum already? Biological databases and stuff? Yeah, Hadi, tell me. Sorry?
Used it once.
Once was it? Okay. Okay, fine. Then what I'll do is maybe after this end of the session, yeah, first I'll just try to get through some of the content, and then maybe I'll open the browser, okay, and just show you some of the things that you can do with the database. Okay. So so why I bring that up is, so this particular sequence has been downloaded from the NCBI database. Okay, and this ID is corresponds to that. So hence, you do not have a CHR6, that is like, let's say how we denote a chromosome 6, but you have another ID. Okay, so this ID of this particular sequence is your chromosome colon followed by start and end sequence of the chromosome. Okay, and so in a FASTA format, this is what it means. So you have a header from greater than sign followed by your sequence. Okay, so like this, within one file, FASTA file, you can even either have one sequence, that is one pair of header and followed by sequence, or you can have as many as you like. Okay, you can have thousands or millions of different sequences as well, but all of them have to follow the same format. That is, first a header starting with the greater than sign followed by your sequence. Okay, and the sequence, it can be also inserted in two ways. Either you have all the complete sequence in a single line, or you can also break it in and have it in multiple lines. Okay, so the way this format works is, after the greater than sign, we say that the sequence of this particular ID starts, and it goes on and on until you see either the end of the file or the greater than symbol. Okay, so everything from after the greater than line till the end of the file or the next greater than line is the sequence of this particular header ID. Okay, everything clear till now? Yeah, any questions? Good. Fine. Now another one, so this is like, FASTA is a very generic format. So you, in FASTA format, basically, as I mentioned, so you can have protein, you can have DNA, you can have anything, okay? And from any source, so, CDA, cDNA, etc., whatever. Now another format, and even the reads that you sequence also, yeah, from the sequences that we had. But the one format which is now specialized from the sequencing, yeah, so remember in the first day, we had gone through like some of the sequencing technologies, and there I mentioned like the sequence. So we give there are machines where we give our input, which can be in the form of a sequencing library, which can be built from either genomic DNA or cDNA, etc. Right? And we give that library to the sequencer, and it generates the sequencing data and give it to us. Right? Now, how does the data look like? Okay, so that data in most of the cases, except for just PacBio and Nanopore, okay, is in the format of a something called the FASTQ format. So FASTQ is basically nothing but FASTA format plus the quality values. So hence the name FASTQ. Okay, and so this format, the way this format is, the data is stored slightly differently than your FASTA format. So here, each read, yeah, so every fragment from the sequencer is read, yeah, and then you get that particular sequence. So each read comes in the form of 4 lines. Okay, so the 4 lines are, first starts with the @ and followed by the ID of the read. Yeah, so the whole thing after the @ symbol is a read ID. Okay, so like in the FASTA format, we had something starting with the greater than symbol, and then that is the ID of the sequence. Similarly, in the FASTQ format, you have something like the @ symbol, which denotes that the whatever is following to @ is your read ID. So the next line is your complete sequence. Okay, the sequence of the read that you have, then followed by a separator, which is something called as a plus symbol. So if after the read sequence, you have a plus symbol, that means that till now, here the sequence is complete. And now after the plus symbol, this is optional. So you can either have the same read ID repeated after the plus symbol, or in most cases, not. Yeah, in most cases, you can just have a simple plus symbol also. Okay, so what do we have? First of all, identifier, then the sequence, followed by a plus sign, and then may or may not be the ID. Okay. What follows next, the last one, is the Q component of the first Q, that is the quality scores. Now, if you remember from the first day, I mentioned that the sequencer determines that what is the nucleotide on a certain position based on the color intensity, right? So we have these different chlorophores where we have A, T, C, and G, correct? So how the sequencer determines this is based on the color intensity that it receives from the laser. Yeah. So like, for example, you can think of that we have cameras over there, right? And as soon as a fluorophore is released, so the camera captures that color, okay? But how do you capture that color? So you actually capture based on a certain intensity, right? So let's say once you have red, but the red can also have a slightly varying intensity also, okay? So what happens in the sequencer is it uses this value of the intensity and calculates something called as a quality score, which we also call something called as Fred quality score, yeah, which basically is the confidence with which the sequencer says. So for example, in this particular case, you have a T. So E here denotes that E is the quality score of this particular T. Like this is the confidence. So whatever is the value corresponding to E, so that I'll touch upon after this, right? So E, the value of E is the quality of T, and quality of T basically reflects that it is the integer representation of the probability that the corresponding base call is incorrect. Okay. So what is the possibility that there has been some sort of a inaccuracy in calling it a T? Yeah. So this is how it is calculated here in the form of a quality score. And here now, if you notice, the number of bases in the sequence and the number of letters in the quality score, they are same, because every base would have a corresponding quality value, which is denoted by these alphanumeric characters. Okay, everything clear till now? Any questions? Okay, that's good. Okay, now the way these characters are, so now how does it, so that this particular Fred quality score, it normally ranges from 0 to 50. Okay, and so how this score is converted, so I'll come to that also. But first about the scoring. So if a Fred quality score of a particular base is 10, that means that there is a possibility that 1 in 10 times that the base is called incorrectly. That is, but the confidence with which the sequencer says that it is a T here is only 90%. Yeah, and in terms of sequencing, like these days, 90% is quite low. Okay, so ideally, we generally have Fred scores ranging between like 25 or 30 or even beyond that. Okay, so this is how, let's say, it roughly translates to, right? That 10 means 1 in 10, and 20 means 1 in 20. And this is like, you see the formula over here, right? So this is how they calculate the probability, right? Again, you don't have to memorize any of this formula, right? But just, this is for your understanding that how the quality are represented in this. Okay, and now how do these letters correspond to the respective quality values is based on the SCHI value of these letters. Okay, so if you know that in computers, right, every letter you have on your keyboard, okay, let's say A, B, C, D, E, F, also uppercase and lowercase as well, right? All of them have something called the SCHI value associated with them. That's SCHI value, ASCII. Okay. Now everything has a SCHI value, and this is what the FASTQ format utilizes to encode the quality scores. Okay, and the way they are encoded is because, like, you get a range of A, B, C, D, F. So for example, uppercase A, it has a ASCII value of 65. And the way the values are encoded is Fred quality plus 33. So sometimes if you see, you would see Fred quality plus 33 encoding. That means if the ASCII value is 65 and it follows a convention of Fred quality 33, so the actual quality score that you have is 65 minus 33. That is 32 over here. Okay. And what is the probability of this? So this is how you calculate the probability, and that's what it boils down to. So 32 is roughly close to like 30. So you have, like, 99.9% accuracy of the base called over here. Okay. Now, any questions till now? Is this clear now? Yeah. Again, you don't have to memorize any of this. Like, most of the tools, right? So they automatically detect this quality score and take care of it, because, like, this quality score is bit important in some of the things. For example, when we would be doing the QC step today, okay. So that time also we will evaluate and see, like, how the quality scores are looking for our data, and then we will see if there is a need to do the trimming or some sort of these things as well, but which we would still do as a practice. Okay. But these are the things, which are also used by all the bioinformatics tools, the quality scores, when they are trying to process these files. Okay. Yeah. So this is one of the most important formats for your sequencing. Yeah. Now, once we do the sequencing, right? So we get data in FASTQ format, and once we also do any sort of processing, for example, we do quality control or trim the adapters. So if you remember, on the first day I mentioned that sometimes you can also have adapter sequences sequenced as part of the sequence, which ideally should not be the case, right? So we also have tools which can then detect these sequences, yeah, and trim them off, yeah. So let's say, for example, if the tool detects that this is the adapter sequence, then it will cut them off. And after doing that also, the data comes in a FASTQ format, the same format. Okay, it's just that it is trimmed, or it is filtered for where it removes any low quality reads. Okay, so what this comprises is, so these 4 lines together in a sequencing context mean that this is a single read. Okay, so in the sequencing you read the DNA, right? So that read, so we every time we talk in terms of number of reads. So remember how in RNA-seq I mentioned we will sequence 20 to 30 million reads, right? So this is one of the read, how it is represented in the FASTQ format. Like this, if you have 20 to 30 million reads, so like this, these such 4 lines would be repeated those many times with, of course, a different sequence, different ID, as well as a different quality score. Okay, everything clear till now? Any questions? Yeah, Saida.
So I understood how, based on the ASCII value, this is like the read is encoded. But then in the first place, how are the quality scores actually assigned to each base? Because we see the characters here, E, F, C, all of these. So like, how do you initially assign those characters?
This is the sequencer. So sequencer determines based on the intensity. So for example, let's say let's say T, yeah, for let's say, for the sake of argument, we have T, and it is encoded by the color red. Okay, and whenever a T, in this case, when a T nucleotide would be added, so the red fluorophore would be released. Okay, but the intensity, so when the camera takes a picture, right? So the intensity of the red can vary when, let's say, you get sequencing of when this T is added, or this T is added, or this T and this T, or so on. Okay, so for example, this T, if you see, right? So this T has a different character than this T, and even the second one as well. Okay, so why we have this? Because the way the camera determines is, so this is like, I'm just so I'm not talking about pure technical, but this is roughly how it estimates. So let's say if I get a very bright red, yeah. So for example, this kind of red, I would say yes, I am 100% sure that this is indeed red. Okay, and let's say, so I'll come back to the format again. Now you see this red, right? Or rather, you see this red that okay, laser pointer red is same as this one. So you see now this red, the laser pointer red, and this red is different. Okay. Now these both color can come from the same nucleotide added. That is, a T produces red anyway. Okay. But what kind of red it produces? So that can, because of various factors, the intensity of the red can vary. Right. Now the color is the quality is determined based on this intensity. Okay, so let's say I say if it is a very bright red, and that means that it's 100% sure, or let's say 99.999% sure, because first of all, the sequential quality score, if you see, it's never 100%. Okay, because of several factors, like you can't reach 100% accuracy in sequences. It always has to be like some sort of a error rate would be there. It could be 0.00001, etc., but there would be some, let's say, kind of error probability over there. Right now, the way that is calculated based on like this, based on the color intensity. So if let's say you have a bright red, you will have, let's say, with the highest red quality, and let's say if you have like such bit dim red, okay, so the intensity would be lower. So then the sequencer will like, yeah, maybe, yeah, probability is red. Yeah, so that is how it. This is the kind of intensity it uses to convert that into your quality. Okay, clear? Anybody else? No? Okay, fine. Then let's move on. Yeah. So, so what we have touched till now. Yeah. So we have the raw sequencing data, which comes in your FASTQ format, and then once we also process it, then also we have in the FASTQ format. What is the next step we do in RNA-seq? Anybody remembers? After removing the adapters or trimming, what is the next step in analysis? Any guesses? What do we do? Yeah. So we have now cleaned up the reads. We have good quality reads over here. Any guess what we might do next? Exactly. Yeah. So next step that we do, thank you, is we align the data. Okay. Now, when we align this data, then that data that we get is there in something called, we have another sequence format for this. So that is the SAM format. Okay. So this is, I think, sequence alignment format. Okay. So the way this particular data, the aligned data is stored. So when we are aligning the data to your reference genome, okay. Now think, so before looking at the slide, think what all information you could have when you are trying to align a read sequence to your genome. Okay, anybody, any guesses? Like what you might, what kind of information you should ideally have? Yeah, okay, don't look at the slide. I've hidden the slide now. Okay, not that you don't have, you already have the slide, but still, what kind of information are we trying to see? Like we have a read, we are aligning to the genome. What are we trying to find? Anyone?
Sir, maybe mutations.
Yes, that's one. But how do you something more? Matches, sure. What do you mean by matches, Pratima? Do we know where this read is coming from in the genome till now? Based on the format that I showed you till now, the FASTQ format, for example, do we know where in the genome it is coming from?
Sir, we have the chromosome number and the ID, right?
Exactly. So we don't have, no, we don't have that yet. No, we do not have that. Okay. Oh, before actually moving to the next format, so just one small note on the ID on the FASTQ format. Okay. So you see this ID, right? So it looks very complicated, no? But actually it is like quite simple, because I'll tell you why. So I'll try to break this down, the ID. So this ID is coming from the Illumina sequencer. Okay. So if you have to break down this ID, so the way you would try to interpret it, again, nobody reads it. Okay. It's just for your understanding that what actually a read identifier represents for Illumina in this case. Okay. Again, as I said, nobody has to remember this, because all the tools they take care of this automatically. So the first bit after the @ sign is like HWI, etc., till the first colon is the ID of the sequencer. That is, so this particular data which is coming out of from, so it is coming out from a certain sequencer. Okay, so one physical machine which is provided by Illumina. Now each machine which Illumina has provided all over the world would have a unique identifier. Okay, so like in the laptops, for example, you have a MAC address, no, which is like unique to your system. So no other system in the world can have the same MAC address, right? So similarly, Illumina also has released like different IDs. So for example, you see HWIEAS209. So this is the ID of the instrument, of the Illumina instrument that was used to generate this data. The next 0006, this refers to the number of sequencing runs. So for example, we could do sequencing again and again several times different samples, right? So what this denotes is that this is the 6th run on this particular sequencer. Okay. Next is the flow cell ID. So you remember I showed you that there is a glass slide which has like multiple lanes, and that is where we load the data to sequence a sample. So that also has a unique ID. So all the glass, so the flow cells basically provided by Illumina are unique. You have a unique flow cell ID attached to it. So the two flow cells in the world by provided by Illumina will have the same flow cell ID. Okay. So what do we have? ID of the sequencer followed by the number of the run, like what is the number of run for this, followed by the flow cell ID. Okay. Then followed by colon, a number which is between 1 to 8. So this 5, any guesses what it could be? So what do we have in a flow cell? In a flow cell, we have lanes.
Lanes.
Exactly. Yeah. So this 5 represents that this data is coming from the 5th lane. Okay. And then now these 3 numbers that we have. So now each lane further is also divided into a matrix. Okay. Now this 58 actually is the ID of one of the metrics in that flow cell. So basically it's just like a grid. Okay. And then this last 2 numbers, 5894 and 21141, these are the X and Y coordinates in that particular location. Okay. You see how very meticulously, like the complete information of where exactly this read is coming from in the flow cell is stored in the read ID, right? Which sequencer, which run, which flow cell, which lane, which particular matrix, and the X and Y coordinates inside that matrix. Okay. Then followed by, we have a hash, and then this is something called as the index. So remember I mentioned about the pooling of the samples, right? So I'll have like, one slide later where I'll, explain you a little bit like what do we mean by pooling. So this basically the 6-digit nucleotide refers to the index for this particular sample. Okay, so in sequencing, you can use indexes, barcodes basically, to pool multiple samples and sequence it on the same lane, right? So remember where I mentioned that we can have, let's say, 100 million reads for a lane, but we only need 25 million reads for one sample for RNA-seq. So it means we can sequence 4 samples in a single lane, right? But how do we separate that out is based on this barcode sequence, which we add in the starting before sequencing. Okay, so this read ID also comprises of that. All right. Any questions now? Okay, fine. Then move on. Yeah. So the next step. So coming back to the alignment format. So for alignment, right? So what do we know? So in this format, anywhere, did we see where this read is coming from in the genome? Anywhere? No, right?
No, sir.
So exactly. So for that, what we do, so this is actually what alignment is doing basically. In aligning the data to the genome, this is exactly what we're trying to do. Yeah, the most important thing, which particular chromosome this read is aligned to, and which particular position in that genome. Okay. So the way this format is first of all stored is first in the when you open the file. Yes, so the SAM file also you can open like normally, but ideally not, because such files they can be big. But there are tools to open these files as well. So the first few lines, they start with the @ symbol. They contain the header. So the way this header is like we have first of all a header line which starts with the HD, which says that this is the version 1 of this format. So within SAM format also, like of all softwares, so these software and the format versions can also change. Yeah, that let's say today we have that SAM version 1.0, where everything has to start with the at the rate, and we have like certain codes that how every information would be stored. Let's say later on, somebody comes up with the idea, oh no, the current format is not correct. We need to have an upgrade. So that time we can have a different version for this. Okay. So in this particular case, as of now, we have like let's say version 1.0, and then it says SO. That basically means sorting order. So we can see. So this tells you that the whatever data you see at the bottom is the data sorted in any format. So for example, in this case, we see coordinates. So that means all the reads, which I'll talk about shortly, all the data is sorted in the base of the coordinates. So like you have chromosome 1 coordinate, let's say if it is, in this case, it is 60,000. Then we would have, so this is the first read which is sequenced, aligned. So this means that there no read has been aligned before 60,000 base pairs on chromosome 1, right? So like this, you have the sorting order. Next, let's say you have all the length of all the chromosomes which are present in your genome and in your, let's say, the reference genome that you used. So in this particular example, it just says chromosome 20 and the length of it. Okay. So this means that we align this data to only chromosome 20 in this case. Okay. Next is the PG. So PG basically is used to store that which tool was used to generate this particular SAM file. That is, which particular tool was used to align the data. So in this case, we have something called as a top hat. Yeah. So this is actually like a bit old, so you don't have to worry about it. We will not be covering a top hat in this internship. But it's just for an example. That PG is basically used to store this information. So you see like how header is used to store a lot of information, which also could be very critical down the line. Yeah, that is like how the data is sorted or not, what is the length of each chromosome, and then what tool and exact command was used to do this alignment. Okay. So next in the next session, I think when we would be aligning the data. Okay. So that time also I'll show you like the command that we will run, the same command you will see in the SAM file which would be produced. Okay, so then it would be more clear. So like this at the using at the rate sign, you can have like multiple other fields as well. Okay, but these are, let's say, the ones which are like most important ones. Okay, now after we have all these at the rate lines, starting with at the rate, which is basically refers to the header, then we have the actual aligned data, so which is in this format. So all of this is not two lines, it is everything is in a single line. Okay, so what you see here is first the way the format is stored is first is your read ID. So you see the ID that I mentioned over here in the FASTQ format, right? So that ID is in your column 1. Then we have a tab, then called by something called as a flag. Yeah, I'll come to the flag bit later. Okay, then followed by the chromosome to which this particular read has been aligned, then followed by the position, the starting position. Yeah, because you see like this base is the read is very long, right? So you can't have all the positions stored here, right? So what it stores is the starting position where, let's say, the first letter of the this one is aligned. Okay, and now the way we have a quality score in the sequencing data, right? So where the sequencer says confidently or not, like how confident it is to call a certain base. Similarly, aligners also have something called as a mapping quality, where they say how confident they are to say that this read is actually coming from this particular location. Okay, so for that we have something called as a map Q score, that is a mapping quality. And then we have something called as a cigar value. I'll shortly explain you that also. Okay, this is also very critical. Okay, and this might contain your this SNP data. Yeah, so that somebody had mentioned earlier, right? So next, what we have is the 3 columns. Yeah, so which can actually correspond to your this second pair in the read. So for this, just one second. Let me actually pull out one more slide to show you just one more concept. Second. Okay, any questions till now, first of all? Yeah, just stop sharing the screen for a second. Okay. So here actually I thought like I had covered this, but not. Okay. So this is one small concept here, which I also wanted to cover, is what is something called as a paired end sequencing. Okay. Now, when we are trying to sequence our data, first of all, are you guys able to see my screen? Can anybody do a thumbs up if you are? Great. Yeah. So in the sequencing, right? So the way we sequence it is, right? So we have a fragment. So what you see in the middle, which is the red and blue, this is your sequencing fragment that we are trying to sequence. Okay. The way we sequence, so we do not sequence sometimes a full fragment of the thing. Okay. The way we sequence is certain bases from one side and certain bases from other side, and that is like enough to determine that where this read is coming from. So we have a concept, something called as a paired end read, where we do not sequence the fragment only from one side. Rather, we do it from both the sides. Okay, so we have adapter sequences on both the sides, right? So the way the sequencer works is, so we sequence, let's say 100 bases from or 50 bases from one side and 50 or 100 bases from the other side. Again, like the length of this reads, they can vary. Okay, now these two reads together, they form what we call as a paired end read. Okay, and now if you notice here in the read ID, right? So I mentioned that I will touch on this a bit later. So you have /1, right? So what this represents is that this read ID is the same. Yeah. So this fragment, if you remember in Illumina, this would be attached to the exact same location on the flow cell. Okay. But because we are sequencing from both the sides, one side and two side, we have read 1 and read 2. So everything else would be same. And then how do you distinguish it is read 1 or read 2? Is by /1 and /2. Okay. So in RNA-seq also, this thing is a bit important, but actually this is way more important in the case of like DNA and RNA, where we are trying to sometimes detect indels. Okay. I'll see if time allows, I might touch upon that also a little bit. Okay. But this is what this read 1 and read 2 correspond to. Okay. Any questions till now? No? All good? Okay. So now coming back to the SAM format. Now these three next columns that you see, they actually correspond to this read 2. So what this means is, so this read ID, what it says is first three columns, so this column that you see, chromosome and position, right, and mapping score, these three are used to represent that where in the read 1 is aligning to which location. Okay. And then the next three columns are similarly used for your read 2. And if you have a star, that means that it is not aligned. Okay. And then we have the next column, which is the actual read itself. So this read is basically corresponding to the read 1, and then followed by the read quality. Okay. Everything clear so far? Any questions till now? Okay, thumbs up if everything is fine. Yeah, Uday, tell me.
Sir, how do we differentiate between read 1 and read 2?
So it's based on this particular ID that you have, like you can have /1 and /2 in the end of the read ID. So when we also download the data, no, later on, so I will see you. So you will actually get two different files, FASTQ files, which would be like denoted by _1 and _2. Okay. So this is how the sequencer stores the information. Okay. So this is there in the FASTQ header itself.
Okay, sir.
Yeah. Okay. Anybody else? Okay. All right. Fine. Now, coming back to one of the very important features here, which is something called as a cigar value. Okay, so till now, based on this information, what do we know? This read ID is aligning to this position, this chromosome, and this position with this quality. Okay, I'll come back to cigar shortly. And the corresponding read 2 is aligning to this, is not aligning in this particular case. Okay, And then we have what is the exact read of the sequence and what is the quality score. Okay. Now, somebody mentioned earlier about SNPs. Okay. So how do we know that how good or bad this read is aligning there? For that, we have something called as cigar string. Yeah. So I don't remember the full form of cigar now, but this basically string, what you see over here, is what is used to denote this aspect of it. So in this case, so these are some of the letters that you can see in this cigar string. So uppercase M refers to match, that is the exact match. Then you have X, which denotes mismatch, and then you have deletion is by D, insertion by N, and gap by N. Okay, so 100 M in this particular case means that we have total of 100 is the length of the sequence. Okay, and 100 uppercase M means all of them match exactly to this particular position. Okay, and similarly, this is an example of another, let's say, cigar string, where it says 1M, which means the first base, let's say in the case of C, is matching perfectly. Then we have two mismatches. So the next two are mismatches wherever it is aligned. Then we have three matches. Okay. So similarly, we can have like, three matches, then 7 ends. So where, what this means is, so the first three bases are aligning, let's say, to one position. Then there is a gap, and then the remaining of them are aligning to after 7 bases in the genome. Okay, like after 7 bases, we have a gap, and then we have 4 more bases which align there. Okay, now those 4 can, let's say this gap could potentially maybe come from, let's say, insertion or deletion, or in the context of RNA-seq, it can also come from, let's say, your splice junction. So let's say you have a read which came from a splice junction, right? So when you align that to the genome, so of course, part of it would align to one part and another would align to another portion. Okay, so this is how this cigar string is used in this case. And similarly, when you have this mismatches like this X and all, so this basically ideally, refer to your SNPs and everything. Okay, any questions till now? All clear? Okay, good. Fine. So now this is how, we have now aligned the data, right? So now I'll touch upon another file format. Okay. Now see what we have done till now. Process the data. We know where this particular data is aligned to, right? Now, next we need to know, so what are we ultimately trying to quantify? Okay. So ultimately we are trying to quantify genes, right? That which gene has how many reads. Okay, so we do not have that information yet. We only know which chromosome, which position, and which I read is aligned there. Okay, now how do we do that? So for that, first of all, before we move on to that, we have two formats which are used to store the gene information. When I say gene information, which means, so what does the gene comprise of? So gene comprises of, let's say, the start of the gene, end of the gene, gene name, gene ID, etc. Right? Then what is there in the gene? So you have sometimes 5' UTR, 3' UTR, exon, intron, CDS sequence, etc. Right? So all of these are part of a gene, correct? So we have some standard formats. These are called GTF and GFF format, okay, which are used to store this information of genes. Like basically we call it annotation of the genes. Yeah. So here, for example, the way this format works is you have like, let's say, chromosome ID, and then you have a source, like where this particular GTF file or GFF file is coming from. Then you have what exactly these next coordinates are. So for example, in this case, you have a gene, and then you have starting coordinates of the gene and the ending coordinates of the gene. Okay. Then followed by you have a dot, and then you have a plus symbol. So this denotes that gene is in the positive strand. Okay. Then you have another dot, and then it has gene ID. So in this case, like this is an ensemble gene ID. Ensemble is one of the databases. Is this being covered already with you guys? Ensemble? Anyone? Has this been taught in the curriculum? No. Okay.
No, sir. Again, some of us are just aware of it.
No, okay. Oh, but you're aware. Okay, fine. Let me see. Like, if I try to find some time and maybe share some resources as well, because these are also sometimes very important, because these are all standard databases. Okay, where all of this information is stored, right? So, like for example, in the case of human genome, so there are databases. For example, like in the case of Ensemble, which stores all these genes, and in this case, they have their own gene IDs as well. Yeah. So NCBI, they have their own gene IDs. So similarly, this particular annotation is coming from Ensemble database. So hence you have an ID which is there from the Ensemble database. And then you have, of course, a corresponding gene name. So this is TP53, which is one of the oncogene, which actually causes cancer. Okay. And then you have the coordinates of the transcript. So transcript is basically the mRNA coming from that particular gene. Okay. So you have the coordinates for those. Similarly, you can have so this particular gene at the moment, it seems like we have one exon. I think it has more. Yeah. So it has this exon information as well. Now this has a bit more information. So you see that gene, transcript, and exon. Now they have slightly different information, all of them. For example, a gene only has gene ID and gene name. A transcript, what does it have? It has a gene name, gene ID, like which gene this transcript is corresponding to, right? So of course, every transcript has to come from a gene, right? So what is in a way the hierarchy? You have gene, which has transcripts. Every transcript has exon and introns, correct? And that can have like further, you can have CDS or not. Okay, CDS and UTR sequences, correct? So similarly, you have a transcript ID associated with it, and then you of course you can have the gene name. Okay. Similarly, for exon, you have like gene ID, which transcript ID. So every transcript would have a certain exon, and then additionally you also have the exon number. So sometimes you can have multiple exons in a transcript. So you can have like this. So like this is how a GTF format is used to store this information. Okay. Similarly, you have GFF also. So it is again bit different format, but like more or less trying to do the same thing. Okay. And okay, any questions so far? No? Okay, all right, fine. So what we will be doing, so ultimately, right now, when we will be using some of the tools, so it uses this format, that is a SAM format, which contains all the locations of the reads, along with this gene annotation format, which contains the location of all the genes. Because if you think about it, these are the only two things that you need, right? Where the read is aligning and where the genes are, the coordinates of the genes and coordinates of the read. So in a way, if you combine them together, so then basically you can calculate what is the number of reads for each gene in each sample, right? So this is what we were doing in some of the tools like down the line. Okay. Now let's get to some of the hands-on part of it. So now may I request you all to open your terminals? Okay, and don't run this yet. Before this, I'll try to do one more thing. Okay, can everybody please open the terminal? Or if anybody has any questions, please let me know.
So can we work in the same terminal where we downloaded the packages earlier, or
Yes,
do we need a new one?
definitely. Yeah, as long as you have this condict activated, it's fine. So you have that bracket base there. If that's there, then it's fine. Okay, let me share my screen and maybe we'll do this together. Because I think first I thought I'll just give you the commands, but let's make the commands in the slides, okay? So that you guys can follow as well, and then we will do this. Do you see my screen, right? Okay, perfect. Okay, so first of all, I'll tell you what I'm trying to do in the next steps. Okay, So what we would be doing now is basically downloading some data from one of the database, okay, and then also trying to process this as well. Okay, by processing, like we will be doing some sort of a quality control and also trimming it, okay, and then checking the quality score. Let's try to do this now. But before that, what I will try to do is, because in the next steps we have tutorial where everything is done in your home directory. Okay, so but in home directory of your WSL, you might not have sufficient space. Okay, so what we will be doing now is we would be, let's say, creating a folder in, let's say, your C drive. Okay, or you can create anywhere, right? And I'll update the slides after this. Okay, so you can create a folder anywhere in your system. Okay, so the main thing that we're trying to do is, so whoever has WSL, for example, so they can install. So for Mac users, you don't have to do this step. Okay, so this is just to take care of the space issue, because C drive would have a lot of space, and by default WSL doesn't store much information in your home directories. So hence it's like, let's say, useful to have it elsewhere. So first we create, let's say, a folder called as Neighborman. It'll be same as the others. Yeah, Neighborman work. Yeah. So first, so basically what we are doing, so we are creating a directory in C drive to run the analysis in. Okay. And next thing which we would do is we would create a shortcut in our home directory in your in WSL. Okay. Now to do this, you can run these two commands. Okay. And for Mac users, only this. For Mac or Linux. Oh, Neighborman has the uppercase. I'll also type these in the chat also, okay? Okay, so for Windows, yeah, for Windows, I do these. So these are two different lines. And then for Mac, do this, yeah. And please let me know, everybody, like when you do it, okay? Yeah. Any questions, any issues till now? Or why we are creating this and stuff? Okay, can anybody let me know who is having any issues or you're not able to do it and stuff? Anyone? And also, once you create this folder, right, this Neighborman work, and again, like, please ensure that you are using the same uppercase and lowercase denomination, because if you do, so then it would be easier for you in the next like rest of the internship, because we have provided all the commands with this particular let's say format. So then you can simply copy paste the commands also, like if you don't want to type everything. So it's important that you use the same uppercase lowercase format that I have here. Okay, can everybody give it okay, thumbs down if you are still doing it? Okay, thumbs up if everybody's done. Okay. Safe to assume everybody's done? Or if anybody has still issues, please, now's the time. Or still doing it. Okay, great. So then if that is the case, it means everybody has done it. So let's move on to the next step. So now what we would do is download one particular data set from one of the databases in NCBI. Maybe I'll see first. Let's try to go through some of the slides, and then maybe end of the session, I will show you some of the databases. Okay, like how do I find this ID? Okay, so first of all, run these two commands. Okay, so first is which prefetch. So prefetch is so remember in the previous session we installed something called as SRA tools, right? So that is what is used to. So SRA is one of the database within the NCBI. Okay, so which we will be browsing through a bit later, which is used to store the sequencing data. So some of the research papers that you might encounter in research journals, wherever the sequencing data has been generated, so they have a obligation where they have to submit the sequencing data in a certain database. So SRA is one such database where the sequencing data is deposited. The raw sequencing data is deposited by the researchers. Okay, and then once the paper is published, it is freely accessible. Then anybody can just go and download it. All right. So we use by SRA, they have provided a toolkit called as SRA toolkit, which we installed using SRA-tools, right? So within that toolkit, we have something called as a command, which is called prefetch, where you just provide the ID of the sample, and then you can download the file. Okay. So now let's do this. First of all, after you have created this directory, can you go inside this directory? Okay, I will not give you a command for that. Like you guys should know by now. Like how do you go inside this particular directory once you have created it? Anyone? Yes, a CD and space the name of this folder. Okay, CD space Nyberman work. And also now in the previous step, we created this soft link as well, right? So for all the Windows users, you would see this folder if you go from the File Explorer. Whatever you're doing here in the command line, everything you can see from your File Explorer in your C drive, Nyberman work. Okay, and for Mac and Linux users, you will see this in your home directory. Okay, so go inside this particular directory and then run these two commands one by one. So which is a command which is used to find the location of a particular tool that you have installed. So in this particular case, we have this tool prefetch, which is part of the SRA toolkits. It will just simply tell you the location of it, just to this is just to confirm that yes, everything is installed and it's there. Okay, you don't have to run this again and again, but yeah, this is just other confirmation that okay, everything is installed and in the correct place. And then to download the data, you use prefetch space ID. So you can just simply copy paste this ID from the slides that you have. And please let me know once you are done. We'll do this in a group manner, okay? Anybody having issues? Please let me know.
Yeah, it hasn't downloaded.
What's the error, Pratima? Can you show me? Wait.
Yeah, I can.
Yes, 1 second. Yeah, try now.
Your
Yeah,
presentation
sorry.
might stop.
Yeah.
You can see my screen, right? I'm just.
It's coming.
Should I full screen it?
Yes, please.
Yeah. Okay. 1 second. I'll just.
Okay, let's give me a second. Yep, I reckon. I think we need to provide some. Wait a second. Yeah, just give me one second, guys. I will give you guys a command. Maybe like use that to download it. Okay, yeah. Can you just, okay, that's fine. So how about this for all the Windows users? So once you are inside this particular location in your WSL, like inside the environment folder, can you copy paste this for all Windows? And for Mac, so Shruti, for you, it would be this. So the curl command is for Mac. And the first one is for Windows. Okay. And once you guys have done this, I will also like update the slides for this. And once you have downloaded it, download is complete, then do this part.
Is that for both Mac and Windows? Or?
The last one, yes. So the tar one is for both Mac and Windows. Yeah. Yeah, good. And yeah, one more. So now you see that the folder name is called, so do ls. You see the folder name there, right? That SRA toolkit.3.3.ubuntu 64.
Yeah.
Right? Yeah. So now do this in your 3.3.0 is for you, right? Can you enter this command? So this is also for everybody. Now, for everybody, just ensure that this folder name. So for Pratima, she has like 3.3.0. So just see like which particular of what's the folder name you have. So Shruti, for you, it would be the Mac one. Okay. So here after this PWD, so just enter the name of the folder that you see in your particular system. Okay, and now Shruti, can you do which prefetch? Okay, and now run the prefetch command. Just go up, up, up. No, I mean the one with the ID. Yeah. Give it a second. Great. Okay. Anybody else still having any issues?
Should I stop sharing my screen?
Yeah, sure. Yeah. Yeah, Hajra, tell me. Do you want to share your screen?
So, sir, I've done that wget, the one you sent for Windows, but mine is stopped till 76%, and it's not going up to that.
Just kill it and do it again. Do Control C. Control C and run the command again.
But what about the tar? The third one you sent?
No. First, you have to download. So the download has to be completed before the tar.
All right.
Yeah. Okay. Anybody else? Any issues? How's the installation going, guys?
So after which prefetch, we should do the prefetch with the ID command, right?
Yes. So first of all, which prefetch, when you do, it should show you the new folder that you have made, this SRA toolkit 3.3 or whatever you will have. Okay. So once you confirm that it is that folder, then you just run the prefetch command with the SRR ID.
Yes, so it's showing.
Yeah. Working now?
Yes.
Pratima, how is it going? Is it downloaded now?
Yeah,
Should
it's
be a
done.
small file already.
Yeah,
It's
it's
downloaded,
done.
right? It's
Yeah.
done. Great.
Sir,
Yes, Nikhil?
sir, for me showing you cannot create the directory. I have created neighbor one work which you have shared the command now, sir.
Wait, can you share your screen now? Yep, now. Sorry, guys. Like for this, we try our best to make it as efficient online. I think still we are doing okay though. For the screen. It's you have already created. It's fine.
Sir, when I do this CD, neighbor
No, first you have to create soft link. No, oh, you have created soft link also. Yeah, I do CD neighbor. CD space uppercase N and tab. Enter. Yeah, is there.
Okay, sir, now it came, sir.
Yeah. See, when I see it comes automatically, magically. How sometimes our teacher used to say auto-magically.
Sorry, sir. Sir, now I have to copy the second link which you have shared. Command.
The first one. The one for Windows. The one with wget.
Okay, sir.
Anybody else having any issues?
So after the tar, the third command we sent, what are we supposed to do now?
The export command. But make sure that the folder name is same that is created after tar. So after tar, just do ls. And then whatever folder name you see, if in your case it is 3.3.0, then you can just simply copy paste the command that I've showed, the export one.
Not create directory.
Okay.
All right.
Yeah. And if not, just change the folder name. I'll also share the slide once again. Shruti, use the curl command. I've written the curl command. No? Everything else is same. Okay. So if curl is done, so then do the tar and then the export. But in export, make sure that the folder name is same because for you, the folder name would be different. You can identify the folder name by typing which command? I won't tell you now. Okay, maybe let's do group by group. Yeah. Okay, group 1. Akshay, Samiksha, Pratima, Bhaumek, Uday. Okay, no question, like nothing about the points. How are we doing? Are you guys able to install it? Okay, Pratima has. Okay, maybe I'll start making notes. Okay. Only give thumbs up if you have installed it. Okay. Pratima, yes. Wait, only group 1. Like, okay, I can't keep track of everybody. Sorry. So Samiksha, yes. Pratima, yes. Akshay, is she here? Yeah. Good. Bhaumek, okay, Bhaumek is not here. Uday. Great. Group 2. Varshini, Ananya, Wahaj, Nawaz. Okay, Varshini done. Good. Ananya, Nawaz, how are you doing? Yeah, Nawaz. Any questions? Or done? Thumbs up if
So it's
done.
done.
Done. Great.
No, done.
Okay. Ananya, are you there? If yes, how's it going? Yeah, you are there. How's it going? Any issues? I think she might still be doing it. I think Bashir is not here probably. Krishna, Siddhisha, Manaswini, Hasini, Lakshmi. Okay, Krishna done. Good. Manaswini done. Good. Pradiksha, Hasini, Lakshmi. Hasini done. Good. Siddhiksha and Lakshmi, how are you doing? I think Lakshmi is not here. Or she is. Lakshmi is there. Diksha? Okay, guys, if there is any error, please let me know. Okay. Okay, group 4. Hajra, Saida, Sanjana, Nuseba. Saida done. Nuseba done. Hajra, Sanjana. I think Sanjana is not there. Hajra is there, I think. Yeah, done. Great. Group 5. Lasya, Harshita, Rameshri. I think Rameshri said yes earlier. Yeah, Mushritanam, Harshita, and Lasya are also done. Who is Shivani Vangar? Is that okay? Bhavani. Oh yeah, there. Group 8. Okay, got it.
Yes, Saurabh.
Yeah, no, it's fine. I was looking for Shivani Vangar, like where she was. She's in group 8. Found her.
Okay.
Okay. What about Mulsthanam? Is it there? He or she? No. Are they? No. Okay. Group 6. Mantri, Pureti, Sadvedi, Pallavi. Both the Pallavis and Sai Krishna. Pallavi Naidu done. Okay, first I'll write you Pallavi Naidu in the sheet, okay? Because then for me it is confusing. Okay. What happened to your other group members, Pallavi? Any clue? What happened to them? Guys. Group 7. Sahiti, Sindhura, Sara, Sadia, Praneeth. Sahiti, yeah. You done or you have a question? Or is still going on? Yeah, feel free to type in. Or if you guys need a couple of more minutes, it's fine. Like, we can give you a few more minutes as well. And guys, like I think what might also work best for next sessions is what I'll do is I will share one single shell script up front, okay, before the session, right? then what I would strongly encourage you guys is please run that command or run that shell script. Okay, I'll send you the instructions as well before the session, so that at least we know that all the commands and everything is running fine. Okay, so you have all the steps properly, so that then actually we can discuss. So sometimes this installation stuff is fine, like this can take a few minutes, but at least for the other steps, so I'll share the step-by-step instructions along with the script, so that you run all of those steps yourself, right? And then we discuss it during the session. Or if let's say anybody had any issues as well, of course I'll go through all the steps one by one in the session as well. But it will be good, like if some of you at least can run this beforehand. What do you guys think about this plan? Would you like this? Thumbs up, yes. Thumbs down, no. Or no comment?
They're clueless.
Okay, guys, what happened? Okay, it's the same people I see thumbs up. Like as I say, Prathima is there, Nikhil asked a question, that's good. Hadi and Hajra also I see sometimes, like Vaishni as well. Shruti also. I think more or less everybody does thumbs up at some point. Also, please feel free to provide feedback. Yeah, if let's say you don't like this teaching style or you would like something different, okay, please use the feedback links that we provide in the end for this purpose. Okay, or feel free to let me know even now. Like I'm happy to change ourselves, like whatever makes you guys comfortable to learn. Okay, coming back. Everybody installed or downloaded? Any issues now? Okay, I'll assume that everybody is. So a couple of more minutes. Anybody else having still issues or still downloading? Please let me know now. Shruti, what happened? Shruti, Nikhil. Okay, one by one. Who wants to go first? Nikhil, yeah, tell me what's going on.
Sir, the link soft link has been not created. It's not working.
It just created, no? Can you share your screen?
Sir, I pasted the command, exact command here. Which
wait, show me your screen.
Failed to create soft symbol.
Okay. And read through the whole line.
Sir?
What does it say? Read the whole thing. Yeah.
LN minus S.
No, that's fine. Failed to create soft link. Why? See, what are the last two words?
Home, Nikhil, neighbor one. Okay, this file doesn't exist.
File exists. It's already there. And you are already in that folder.
Sir,
so what you have to do? No, you're there. It's Nikhil. You have to download it now. So run this wget command.
Sir, which command, sir?
Wait, wget. Oh, sorry guys. I didn't see that there were so many messages. Apologies, guys. I didn't see. So just 1 second, guys. Okay, first, Nikhil, I'll help you out. So okay, run this. So just paste exactly what I'm giving you. Okay, first do these two steps. Okay, sorry. Next, Hadi, are you done?
Sir, I have to go with this.
Yeah, do these two steps.
Okay.
Hadi, so everything done, right? Good. Oh, sorry, guys. Okay, guys, if sometime like there is a fraction of second because sometimes sometimes the chat might be in the background when I have the presentation open, so maybe that's why I missed the message. Okay, so Hadi, you are done. Who's next? Syed also said yes. After export, is it which we face? Yes, Hadra, that's correct. So Hadra is done. That's good. So there is Sahiti. What's the issue? Pallavi, what's the issue? Sahiti, what's the issue? Please feel free to share your screens and let us know, and then we'll make you host. Okay, maybe I'll make you host anyway. Sahiti, you go now. Sahiti, your turn. Pallavi, I'll also make you host, and then after this, after Saiti, you can also share your screen. Yeah. Oh, great, Pallavi. Can you full screen it, Saiti? Okay, go down. Keep going down. Down to the end. Do df minus h, df space minus. Oh no, it's fine. Like you already have that. So df space minus h. Yeah, let's try that first. Oh, did you not create this? Oh, can you do cd dot dot and do ls minus l? Cd space dot dot space and ls minus l. No, after, like, first you have to enter cd dot dot enter. So you come out of the directory and there no ls minus l. I just want to see like if the soft link is created. Nikhil, this one. ls minus l. Enter. Okay, so soft link is created perfectly. That's good. So go back to neighbor one work. CD. You don't have to scroll up, just do CD. CD space uppercase N and tab. Tab. Enter. So now everybody, like, now you're trying to see, right, like how the tab is working. Everyone, correct? Now run the prefetch command, Saiti. The prefetch space SRR command was there, no, in the slides. I'll type it in the chat also. Okay, once again. Yeah, can you type this? Good, it's working. Saiti, you can stop sharing. It's downloading now. Let me know if there's any issue. Yes. Thank you. Anybody else? Nikhil, what's happening? Saiti, I'll still keep you as host. Like if there's anything, like just share your screen. Okay. Nikhil, yeah.
Sir.
Yeah.
Hello?
Share your screen.
1 minute, sir. Sir, I think it's done. I pasted that both two comments and it downloaded.
Yep, that's good. And now, yes. Yeah, now run the export command. I'll share it. Okay.
Okay, sir.
Run this export command and then run this prefetch command that you don't need to do which, just run the command right in the end.
Sir, one I have to export, then another one is path. That's it, right?
No, same line. Everything is a single line. The whole thing.
Okay, sir.
And guys, I have like updated the slides with all of this as well. So I'll be just like replacing the PDF that we have uploaded in the drive. Now run the prefetch command, the prefetch space SRR thing.
Sir?
Go to the chat.
Okay, sir, 1 minute.
The previous, yeah, exactly. Yep, there you go. Yeah, that's fine. Anybody else having any issues? Or still downloading? Yes, Shruti. You want to share your screen? 1 second. Let me make you host. Go for it. Shruti. Wait, Shruti. First, I think you need to have the SRI toolkit actually inside the Nyberman work. So what maybe I would propose for you to do is the following. Run this command, okay? So you have to actually move the folder that you had SRI toolkit, okay? So I will type the command, just run this to move the folder inside the Nyberman folder. Here. Can you paste that? Good. And now I will give you an export command. Run that. Run this. And then run this and let me see the output. Which space prefetch? No, once again. No. Oh, sorry, wait. First go inside the directory and then run the export command again. Up. The three ups. Yeah, enter. Again 3 ups and enter. Yes. Now do the prefetch and SRR thing. Prefetch SRR 1553606. Yep, working. Yeah, you can stop clearing. Okay, anybody else? Who else? Sahiti, yes, you want to share your screen? You're still host. Just run the command again. Wait. Yeah, run the command again. Just do up arrow. You don't have to copy again. So you use up and down arrow to move to previous commands, okay? Scroll through the previous commands. Perfect, done. Okay, looks like that was just a network issue. Okay, anybody else who's there? Shruti, I think it was downloading, right? Nikhil, everything fine now?
Yes, sir.
Okay, Saiti, you can raise down your hand if you still have. I think I'll just lower your hand. Just raise your hand again if you have any questions. Anything else, guys? No? Everybody's fine? That is good. Then let's move on. Okay. Let's try to at least run some commands today. Okay. The other thing from next time, this installation issues and all, hopefully we have solved today. Okay. And from next time, I'll just give you some shell scripts up ahead that we can just go through. And let me know if you would not like that. Okay, if you would prefer something else. All right. Okay, so good job. So what we have done till now, right? So we have first of all installed SRA toolkit, right? So sometime like this what we did. So there could be some sort of a version issues as well, but that's fine. Like we have now resolved it. And using this prefetch utility, we have downloaded one of the sequencing data. Okay. Now next, what we would do is just do ls in your folders. Can anybody try to do ls in their respective folders and show me like what you see? And can you just share in the chat, or if anybody volunteers, does anybody volunteer to share their screen? Hajra, would you like to maybe share your screen? We can use that as a demo.
Yes, sir, sure.
Okay, sure. Yeah, thank you. I will just make you host. Okay.
All right.
Yeah.
All right. Just a moment, sir.
Yeah, sure. Take your time.
Is that visible?
It's coming. Yeah, can you maximize the terminal?
Yeah.
Yes. So do you see that folder SRR? So everybody, I think when you do ls inside your folders, you should see this particular folder SRR1553606 in your directories. Is there anybody for whom this is not the case? Anyone? Now's the time. Nope. Great. So it means, so now go inside this directory, Hajra. Like you guys also, let's explore a little bit. Okay. So what's happening? So go inside this particular directory. How do you do that?
CD and directory name.
Exactly. Very good. Do that. Yeah. What's the direct name of the folder? The data where it has been downloaded? The ID name, yeah? That SRR ID. No, SRR. You want to see what we have downloaded, okay? Just go inside that directory, CD space name of the ID. Yes, yeah, you can just do tab. You don't have to type the whole thing.
All right.
Exactly. And now do ls.
Mm-hmm.
Yeah, so everybody, so what you see now, so from the SRA database, right? So remember one thing, okay, this sequencing data, so this can be very large, okay, and generally is. Sometimes you have file in gigabases, or sometimes, in very rare cases, it can even be in terabases of sequencing data. So what this NCBI SRA database does is, it stores all of this data in a bit of a further compressed format, and that is something called the SRA format that you see over here, okay? But what format did I tell you earlier that how the sequencing data is stored? It is stored in a FASTQ format, right? So for this, what we do, we convert. So there is a way how you can convert your data from this SRA format to your FASTQ format. Okay. Now to do that, go inside the Neighborman folder. Okay, Hajra, come out of the folder and stay in the Neighborman folder. And everybody run this command.
I'm sorry, I wasn't mindful. I was named.
Yeah, sure. Yeah. So you have to run this command.
Which one? Do I have to like move back?
Yes, move out. So everybody go inside the neighborman folder and then run this command.
Okay.
So what this command is doing, so again, guys, all of this is in our slides anyway, like what we've shared, okay? And I'll also be updating it, so you don't have to remember all of this. But still, I'm trying to make you guys comfortable in running all of this by yourself as well. So what this command is doing is, it is converting the SRA format of the file into your FASTQ format, okay? And now try to understand this format. Have you pressed enter, Haja?
Not yet, sir.
Yeah,
Oh,
press
yeah, I
enter.
did.
Okay. Yeah, now you did, right? Yeah. You see, so this is it has already now made it. So now do ls and see what's happening. What happened there? Anything new that you see? You see a folder called FASTQ files, right? So that is a new folder which has
Yes,
been
sir.
created. Okay. Now, one more thing. So if you want to see what is there in the FASTQ file, you don't, all of you, you don't have to go inside the folder. Okay. You can also use the ls command to view the contents of the folder. So to do that, what you do is do ls space FASTQ underscore files. So for that, you can just do lsf and tab. Just press tab, it will autocomplete. Yes, and enter. So now what do you see? You see the FASTQ format. Okay. And you see two files, _1 and _2. Remember, I showed you what is there. There is something called the paired end data. Correct. So here you see that we have R1
Yes,
and the read1 and read2 files of the same thing. Okay. Now go inside this folder, FASTQ files. Is everybody following? Is everybody doing the same things that Hajra is doing here? If any issues, please let me know. Yeah. FASTQ underscore file. Yeah. Enter. Yeah. And now let's look at how the files look like. So if you remember, so there was a command to view the first 10 lines of the file. Do you remember which command it is? Anyone? Anyone?
Head.
Exactly. So thank you.
Head space
Head.
Yeah.
Yeah. So do head and space any file name. So you can do either _1 or _. Let's do the _1 first. Do head space file name. Do S and just do tab. Tab once and it will autocomplete till _. Yeah. So now do 1 and then tab again. So you see like this tab, right? Do enter 1 and do a tab again. So like this tab, you autocomplete basically what is already there. Okay. Hajra, do
Head
enter
and
1 and tab again. You see now it autocompletes. Basically, you don't have to type the whole thing and press
Okay,
enter.
all right, got it. Okay.
Yeah. So you see, so these are first 10 lines. So does this sound familiar, everybody? This is similar to the FASTQ format that we saw earlier. Okay. Of course, the ID has changed a little bit because that is how the NCBI database stores this. It changes things a little bit. But what you see, the format is same. You start with the @ symbol, which has the read ID, and in this case, it also has the length of it. Then followed by the next line, which is the sequence, then followed by the plus sign, and then the ID is repeated. And then you have another line which contains the quality scores. Okay, now Hajra, do the same thing with the _2 file. To and tab. Enter. Yes. So these are the corresponding. So basically, so you see that the read IDs are same. Okay. But in the file name, we have _1 and _2. So that basically corresponds to the read1 and read2. Okay. Everything clear, everybody?
Can you
Yeah.
Can you please repeat the part again, so I didn't follow my
The last
internet.
one. Okay. So you remember we had this paired and sequencing data, like how I had shown you earlier.
Mhm.
Okay. So this _1 and _ corresponds to that. So _1 file, it contains all the reads from the read1 component and _2 contains the reads from the R2 component of it. Okay, remember how the fragment is sequenced from both the sides in the slide that I had shown you today?
Yes, sir.
Yes, so this is how it is stored. And now let's see. So ideally, the number of lines or number of reads in both the files, it should be exactly the same. Okay, so for this, there is a command which is used to read the number of lines. So just run this command, wc space -l. I've also typed it in the chat.
Okay, I'll
wc
just
space,
this.
yeah. This is a command to read the total number of. So we will be doing this in the QC as well, but we'll do that in the next session. But today I just wanted to show you. Enter. So we will be running a tool called FastQC, which we installed today. Yeah, just give it a second. So this is the total number of lines which is present in this particular file. It should be identical in both the cases, because obviously these are read pairs, right? So hence the number of reads in R1 and the number of reads in R2 would be the same. Okay. Okay. Any questions till now? Any issues?
Sir, can you share this fast command again?
Fast command, which one? The faster queue dump. It is there in the slides that we shared.
Okay.
Yeah, the faster queue dump one, right? Yeah, Hajra, you can stop sharing now. Let me share now.
All right, sir. Thank you.
Yeah, thank you. Yeah, thank you for sharing this thing. Okay, can anybody, so you're able to see my screen, right? So you see, so all of these slides are there in your script, okay? So see, all this slide is there. So for example, where, first of all, you create these files. So, you don't really have to create the folders. They're more or less created automatically. And then you run this command where you have this faster queue dump, which converts basically the SRA into the paired and fast queue formats that we showed earlier, okay? And then you can visualize the files like this. Okay, and then I showed you that how you can do head for, seeing the top and bottom things for these files as well, right? Okay, so now what I will do is, I think because it's time already, so maybe I'll stop now for the today's session, but I'll give you guys a little bit of exercise. Okay, see, now you guys are very comfortable, I reckon, with working through the file folder structures. Okay, now you see you have the slides already, and you have the steps for fast QC, for example. Okay, so first, you don't really have to worry about the installation. So installation is already done for fast QC and fast P, also for multi QC. Okay, what I would like you to do maybe before the next session, or if not, we'll also go through it. Run these commands. Okay, just follow these steps and go to your respective folder. We have screenshots here as well. Go to your neighborman work fastq folder and run this command over here. Okay, all the steps are provided to you guys. Okay, make a folder for fastq results and run this, and then this would create HTML files. Okay, and because it is present in the C drive, right? So you can also go and view the HTML files in your Google Chrome. Okay, and similarly, you can also run some of these commands that we have mentioned, right? So just try to have some fun with it. Let's see, if how comfortable you are, running these commands. If not, we will cover this anyway in the next session as well. Is that okay for you guys? Is that a good plan? Thumbs up? Yes, no? Okay. Okay, yeah, fine. Yeah, let's, Bhavani, with that, let's wind up this particular session. Okay.
Okay, sir. Fine. Dear students, feedback form has been shared in the chat. Please do fill it out. And we have important announcement regarding next session. As you all know, tomorrow we don't have any session. Our next session is scheduled on Thursday, 26th As per the guide which we shared, the schedule is around 10 to 12, but there is a time change. We have been shifting it to 7 to 9 Indian time, 7 to 9 p.m. Indian time. Is that fine? Works for you all? 10 to 12 schedule has been shifted to 7 to 9. Okay. All right. Saurav, is that fine?
Ma'am, is it only for
Ma'am,
26?
is it only
Yes, yes. Only
Yeah.
for this. Only for 26.
Only for 26th.
Oh, thank
7
you.
to 9, is it?
Yes,
Yes.
7 to 9.
Only for
Okay.
Monday.
Only for 26th, 7 to 9.
All right. All right.
You have other session?
Yeah, the second class which I'm enrolled in, it's on the same exact time.
Same time. Whether that they provide you recording?
Yeah, I have to say how that goes, but yeah, I'll try to
I'm
balance.
extremely sorry.
Yes, sorry, Hajra, this is just for one
Will
session.
be receiving the
Yeah,
record.
yeah,
Yeah, you will
totally
receive.
fine.
Okay, you can just review that. Okay, we have no other way. Please do adjust with that only for one day. Okay.
Sure, ma'am. Yeah.
Okay, yeah, thank you, Hajra.
Well, that's it. Sort of, let's wind up for today then.
Yes,
Is
yeah.
there any
Let's
activity
just close the session.
for the day?
Yeah, I hope there was some productive. Like we
Okay.
had again some installation issues. It was a bit better than yesterday, but yeah.
Okay. All right. Yeah.
Yeah.
Dear students, I guess now you think bioinformatics is overwhelmed. Anyhow, it's all said. Practice make a man perfect. So by practice, definitely you will rule what bioinformatics is. With this, let's wind up for today. And if you have any questions, please make use of Telegram discussion forum. Saurabh will be active over there. If
Yeah.
you find any error,
Even tomorrow as well, yeah.
yeah, maybe you can take a screenshot of the error, you can post it, and you can make use of the forum if needed. All set. Let's wind up then.
Yeah.
Thank
you
you,
guys.
Saurabh. Thank you, participants.
Thank you, ma'am. Thank you, sir.
Thank you.